Ever wondered why protein purification is so critical in research projects? Well, let's dive into that. Protein purification is an indispensable part of scientific research, a process by which a single type of protein is isolated from a complex mixture. Why is it important? The answer lies in the intricate world of proteins. By purifying these proteins, we can study their structure and function with much more clarity. This understanding is the cornerstone of many innovative breakthroughs, influencing various applications in biotechnology and medicine. From developing new therapeutic strategies to understanding the molecular mechanisms of diseases, protein purification plays a pivotal role. It's like finding a needle in a haystack. But in this case, the needle is a protein that could potentially unlock new frontiers in science. So protein purification is not just a process, it's a key that opens the door to untapped potential. Now, wouldn't you want to know how this intriguing process takes place? Dive right into the heart of the matter, the process of protein purification. Our first stop is the extraction of protein. This involves breaking open cells to access proteins within them. This could be achieved through physical methods such as grinding or sonication, or through chemical means like detergents. Extraction not only releases proteins, but also a cocktail of other molecules. Moving on, we land at precipitation and centrifugation. Precipitation is the process of using a salt, often ammonium sulfate, to coax the proteins out of their solution. As the salt concentration increases, many proteins will precipitate out of the solution, allowing them to be collected. This is followed by centrifugation, a technique that uses centrifugal force to separate particulate matter from a solution. The heavier elements move away from the center of the spin, enabling us to separate our proteins of interest. Next, we step into the world of chromatography, a technique used to separate the components of a mixture. There are several types of chromatography, but we'll focus on three main types used in protein purification: ion exchange, affinity, and size exclusion chromatography. Ion exchange chromatography separates proteins based on their net charge. Affinity chromatography, on the other hand, uses a protein's ability to bind to a particular molecule. A column is prepared with molecules that the protein of interest will bind to, and as the mixture is passed through the column, our target protein will stick while others wash through. Finally, size exclusion chromatography, also known as filtration, separates proteins based on their size. The larger molecules pass through the column faster than the smaller ones, allowing them to be collected separately. Each of these steps contributes to the purification of the protein by separating it from other proteins and molecules and allowing us to isolate and study it in detail. Protein purification may seem complicated, but once you get the hang of it, it's as easy as pie. Now, let's recap the key points about protein purification that you should always bear in mind. Protein purification is a pivotal process in research projects, allowing scientists to isolate proteins of interest for further study and analysis. The steps involved in this process, extraction, precipitation, and purification, each play a critical role. Extraction breaks down the cell walls, freeing the proteins. Precipitation separates the proteins from other cellular components, and purification refines the sample to isolate the specific protein of interest. Precision and accuracy are paramount at every step. A small error could skew results and impact the entire research project. So meticulous attention to detail is a must. Ultimately, protein purification is about more than just the how, it's about the why. It's about enabling breakthroughs in research, contributing to our understanding of biological systems, and helping to drive advancements in health and medicine. And that's a wrap on protein purification. Remember, it's not just about the how, but also the why that makes this process so crucial in research projects.